I want to welcome everyone to the OCCPR webinar series. Today, we will be discussing a network exploration tool from the CPTech program called ProNetView. This webinar is hosted by Assistant Professor Dr. Zainab Yunus and her team of the Icahn School of Medicine at Mount Sinai, New York. All registrants are muted, but can use the chat feature, the Q&A, to send questions to the speakers, which we will try to address during the question and answer period at the end of the presentation. This webinar will be recorded and made available on our website. Thank you for joining.
Hi, everybody. My name is Zainab Thomas, and oh, sorry.
Zainab?
Yes.
One second.
Sure.
Thank you for joining the NCI's Office of Cancer Clinical Proteomics Research webinar series. As a part of NCI's Cancer Continuum, OCCPR mission is to improve prevention, early detection, diagnosis, and treatment of cancer by enhancing the understanding of the underlying molecular mechanisms while advancing proteome and proteogenome science and technology development. Through the integration of genomics and transcriptomics with proteomics, also known as comprehensive proteogenomics, OCCPR's aim is to advance basic translational and clinical research through OCCPR-supported programs. This webinar series hopes to educate you to all the resources OCCPR and its researchers have to offer.
Okay, so now Zainab will discuss the tool.
Welcome, everybody. Today we will be presenting to you ProNetView, which is an early version of the tool that we are developing for interactive visualization and exploration of proteogenomic networks. And my lab is predominantly involved in development of interactive data exploration tools that can be explored visually. And Dr. Selim Kalaycı, who's a postdoctoral researcher in my lab, has been the person who has developed this tool and done all the programming. So he will be presenting you the details and he will be presenting you the demo. However, I will be participating in the panel, and if you have any questions, I will be happy to answer, as well as Selim. So with that, Selim, please go ahead. Thank you.
Okay, I hope you can hear me and see the slides now. Please let me know if there are any issues. Okay, so as Dr. Gümüş mentioned, I'm a postdoctoral fellow in her lab at Department of Genetics and Genomic Sciences and Icahn Institute for Data Science and Genomic Technology at Icahn School of Medicine at Mount Sinai. And I will be presenting our work that is conducted under the CPTech consortium that is mainly used for interactive network exploration. So the outline of my talk today, first I will introduce the motivation of our study. Then I will discuss some concrete examples of where our tool can be useful. Then I will perform a brief demonstration, and I will conclude with some of the work that we are currently still working on. So first of all, co-expression networks have been extensively used to characterize the association of thousands of genes and proteins, and these are used to in the identification of new therapeutic targets. And as you may all know, CPTech is generating massive multi-omics data sets, and based on these multi-omics data sets, we build co-expression networks, and we build co-expression networks on different multi-omics data. But these networks that are built end up being quite complex, and they are mostly high dimensional. So we need it was noticed that we would need some software tools that we can use to help better navigate these networks. And for this purpose, we've been developing PronetView, which is a user-friendly web-based interface. And again, it is used to enable interactive exploration of CPTEC networks so that the underlying data within these networks can help in the translation of these results into biological insights and clinical applications. And before we go ahead with the details, is there any need for such a tool? Can't we use existing tools? There are already existing network visualization tools such as Cytoscape. So I would like to briefly mention why we need such a customized tool. First of all, tools like Cytoscape and others are great tools for visualizing networks in 2D, in two dimensions, and they are especially good for moderate sized networks. And they can be used mainly for emphasizing a certain characteristics of your network and sharing the results with your colleagues. Our tool is mainly developed for exploration, for deeper exploration of networks, for each researcher to do their own exploration. And due to the complexity of CPTEC networks, due to complexity and the high dimensionality, we need better, more capable tools to explore these networks in more detail. So, and with the CPTEC specific studies, there are, as I mentioned, high dimensionality, like we have clinical analysis and other metadata such as pathway analysis results. So in our tool, as you will see in our demonstration, we can incorporate them. You can explore those high dimensional data within the same tool. Also, another reason is that, as I mentioned, this tool is developed and customized based on CPTEC data and the user needs. So if there is a specific, for example, data type that is the result of a certain analysis, we can incorporate them and make them available within our tool. Another main feature and our objective in our PronetView tool is to make it easy to access and use. For that purpose, our tool is a web-based resource. That means you can, and it only uses standard technology. So it means you only need to use a web browser. Just use your web browser and go to the URL for the study. And you don't need to install any software plugin or anything, and you can just start exploring the networks. Whereas with, for example, if you are using Cytoscape, you would need to install Cytoscape onto your own local machine and perhaps install additional plugins. And when you move to another computer, for example, you would need to do them again. So it's not very portable, and when you share the results with your colleagues, it's not standardized. If you were to deal with these Cytoscape issues, and you would need to also, for example, import network data into your own system, so you would need to handle all those details. But with PronetView, all you need to do is just access the URL, and you will have the same experience wherever you may be doing your research. And finally, another main reason is that another main feature of PronetView is that it utilizes the third dimension, meaning our networks are in three dimensions, whereas Cytoscape and other tools are mainly by default can handle two-dimensional networks. And when you have such large and complex networks in 2 dimensions, these networks would look like a hairball, whereas in 3D you can easily rotate, zoom in and out, and you can see and you can investigate the networks in more detail. So now at this point, I would like to concretize our discussion and mention our study on clear cell renal cell carcinoma. So we have deployed our tool PronetView on this study. This study was recently published in Cell, titled Integrated Proteogenomics Characterization of Clear cell renal cell carcinoma. And within this study, we integrated proteogenomic analysis of 103 ccRCC tumors. And we built co-expression network using phospholipid data from the tumor samples. In total, phospholipid data included around 21,000 phosphopeptides that are mapping to around 5,800 unique genes. For the network construction, a random forest-based model, namely KRF, developed by our colleagues Francesca Petraglia and Pei Wang at Mount Sinai, was used, and it's mainly capture the co-expression patterns of phospholipid data at the gene level. So that was done basically by making sure that the two genes were connected if one or more phospholipid mapping to the genes were highly associated to each other. So even though the data to infer the networks were used at the phospholipid level, the network was constructed at the gene level, as I mentioned here. And again, this part was conducted by our colleagues Francesca Petraglia and Pei Wang. And as the result of this network construction, the estimated co-expression network involved 3,616 genes connected by more than 11,000 edges. So it was quite large and dense network. Also, based on the topology of the network, we identified groups of highly connected genes or modules. Actually, we ended up with 30 different modules. And we also performed pathway enrichment analysis on those modules, and we identified some of the modules were highly enriched in some pathways. For example, cell cycle and angiogenesis enriched modules were captured using this phosphoproteomics data, which was interesting to, is an interesting finding because we couldn't figure this out using only global proteomic and transcriptomic profiles. This can only be discovered at the phosphoproteomics level. And on the right-hand side, you see a two-dimensional visual of the angiogenesis enriched module. And this is a specifically important network module, and this was discussed in more detail in the Cell paper because it was inversely correlated with BAP1 and chromosome 14 loss, and it was also associated with lower tumor grade. So now at this point, I wanna get back to PronetView and how it is being used within this study and how it can be useful based on these network results. So the PronetView provides two main networks based on the study I just mentioned. First of all, the global ccRCC tumor phosphoproteomics network that consists of more than 3,600 genes and more than 11,000 edges, and also each of the module-specific networks, actually 30 module networks. And within the global phosphoproteomics network, you can interactively explore all the modules, as I will demonstrate in a second, and you can query gene-level information from the global network directly. And also you can individually and in more detail explore each module network. And within these module networks, you can perform deeper querying of gene level information. You can also explore pathways enriched with the module. And also you can query phenotype level information, meaning you can filter for the genes that satisfy a given FTIR cutoff value and a phenotype of interest. And those phenotype Phenotypes are tumor grade, tumor stage, gender, and age. So at this point, I would like to transition into the demo and do a brief demo of our tool. So this is our this is the main page of PronetView we developed for CCRCC study. And you see some explanatory information about the CPTEC consortium and how and the specific information about the CCRCC project. And you see some very brief description about the underlying data that I just mentioned. And at the center of the page, you see a network panel where we display the CCRCC tumor phosphoproteomics network. And you can interact with this network and you can rotate it. You can zoom in and out. Each of these circles or nodes in networks terminology are genes. And the connections, the edges between genes represent co-expression. So as you can notice, it's quite large and certain components are quite dense within this network. And you can also notice that some of the nodes are colored with a certain color. And those colors are indicated on the right-hand side. So as I mentioned, we identified modules, and 6 of the modules were actually discussed in more detail within the article, and we highlighted them in this network. And that these modules are specifically interesting because they are highly associated with certain pathways, biological pathways. And the rest of the modules you can access from this tab. So all 13 module networks are listed here. And if you click on any of them, you can explore each of the module networks in more detail, which I will do in a second. And finally, you see a search button over here, search interface over here, and you can search for a certain gene within the network. For example, I would like to search for oncogene ERG, which was again mentioned in the article, and it was actually associated with VEGF response. If you perform search, you will see that it is highlighted in this network. You can notice module. Very high level about you can also tumor stage see that it is somewhat interesting to look into this click on modules for transit or business and module in more detail. On a module my experience this quite fast. Okay, so it's only for and even though it doesn't many it can get you can notice that some of And by default, gene so that genes are than the others in the network. If you would like, you can switch on all the gene labels and see which genes are located, which genes are found in this module network in more detail. But more often, you don't want to explore this network with all these gene labels, so you can turn them off and just be more interested in the topology of the network. And besides zooming in and rotating, you can hover over each node and this will display again the gene name. And if you click on the gene, you will see that the connections are highlighted. And on the right-hand side, again, the metadata information is populated. So you will see that gene CACNB2 for CACNB2. These are the connecting genes. You see the list of mapping peptides and also, again, the association values with clinical variables. So you can interact with this network in such a way. Another feature I'd like to highlight in this module is how you can make use of this phenotype query option, phenotype query panel at the top. So you see that the phenotypes that are incorporated in our network are listed here: gender, grade, gender, age, and stage. So since this network is highly associated with tumor grade, I'd like to select tumor grade, and you can specify a FDR cutoff such as 0.2, and you can click submit. As the result, you will see that some of the genes, quite a number of genes are highlighted. Those are the genes that satisfy this criteria. So a large number of genes have quite a significant association with tumor grade, and the list of the genes are also provided in this text box. If you want, you can make this FDR cutoff more restrict, and you will see still there is quite a number of genes that are highly associated with tumor grade, and you see the list of the genes provided here. When you are done with your phenotype query, you can click on the reset button, and all the highlights are removed, and you can explore in any way you want. Then at this point, I'd like to go back to the main network, the global network, and do another search. Another gene mentioned in the article was CDC20. CDC20, and this gene was again important because its phosphorylation was found to be representing a mitotic arrest mechanism. So if you perform this search, again, you will see that it is highlighted over here within this violet colored module, and you see that it is also highlighted here. So CDC20 is found to be in module 18, cell cycle enriched module, and you see the same kind of metadata associated with CDC20 populated in this panel. Then I would like to click on module 18 and do another brief exploration there. So when you click on module 18, again, it is displayed in another page. And again, even though it's only this module consists of only 73 genes, it is quite dense, as you can see, and quite a number of genes have high connectivity. And it has the same layout. The whole page layout is the same for all the modules. So, and at the center, you see the network view. And you, on the right-hand side, you have the node level information. But for this module, I'd like to introduce you or explain briefly this panel on the left, enriched pathways. So this is an interactive table of enriched pathways that are associated with this module. So you see they are listed by their name, sorted by their name by default, but let's sort them by p-value. So the most significant enriched pathway is the cell cycle pathway in this module. So if you click on this module name, this pathway name, again, the genes that are associated with this pathway are highlighted in this network panel. And the list of the genes, again, by name are listed in this text box. And you can peruse all the pathways listed on this table. And you can get the list of all the genes that are associated with each pathway listed in this table. And all the other functionalities are the same. You can perform phenotype level search from this panel again, and for each gene, you can display gene level information on the right-hand side. So I will not demonstrate them again. So at this point, I'd like to go back to our slides, the presentation, and conclude with some of the current studies. So we are still developing our tool, and our main purpose is to incorporate other CPTEC networks within our ConnectView tool, and incorporate other cancer types, other data types that may be available, and tumor or normal tissue types. Actually, on the right-hand side, you see some of our preliminary network results, network visuals for our pediatric brain tumor that is ongoing. So hopefully that will be the next to be released. Similarly, we will be incorporating clinical and metadata analysis results associated with each network, so that you will be able to explore multiple dimensions within the same network. We are also continually improving our user interface and adding new functionalities based on some unofficial surveys and interviews with our users so far. But in the near future, we would like to collect more official user feedback via surveys and interviews. And based on those feedback, we would like to add or modify functionalities. And we would like to release each of those resources when they become available and disseminate to the community through social media presentations and events like this. We will be also employing analytics tools to measure user behavior and statistics, so that we can customize our tool and make it more feasible, more effective tool for the users. Finally, I would like to mention that the publication related to this study, and you can see more details of this study soon in the Proteomics journal. It is in press and it should be published soon. But a preprint version is already available in bioRxiv, so you can already see our findings in this bioRxiv version. And I would like to thank you and let you know that we love feedback, and any kind of feedback that you might have, any comments, any questions, any issues, please let us know. Please let my PI, Zeynep Gümüş, and me know, so that we can address them as best as we can. Thank you.
Could you move the ball back to me?
How do I do that again? Sorry.
Yeah. Okay. Thank you very much for presenting the tool. Okay, so now we're going to go over some of the questions that we had in the Q&A. I saw some of them have been answered during the webinar, so that's great. So if you guys could, Zainab and Celine, go over the ones that you want to answer.
Hi, so I could start from the first question. I already answered it in QA, but the first question is how to define the co-expression between proteins with phosphorylation change. Does it distinguish the same up, up, down, down with reverse direction of change of phosphorylation up, down, or not? The answer is actually the random forest model captures the association between the phosphopeptides, both negative up, up, or down, down, or opposite directions. So it's all captured within the model that we employ. And maybe I go to the second question. And the second question is, can you please tell us about the CPTEC Interactive Network Exploration Portal? Can it be studied on infectious viral protein profiles? So the network exploration portal that we have developed is specific to cancer networks within CPTEC. It's specific to tumors within CPTEC. However, the software that we have developed and the infrastructure we have developed can be directly utilized in visualizing infectious viral protein networks. And also the random forest model can also be applied to develop these networks, and then we can apply our visualization. So we have the whole infrastructure to be able to address different kind of questions. And we are actually doing that with another consortium. So these are the two questions I have. If there are any other questions, please let us know and we'll answer them.
All right, thank you again for presenting the tool to everyone. And thank you everyone who has registered for participating in this webinar.
Thank you.
Thank you for joining the Office of Cancer Clinical Proteomics Research webinar series. Please visit our online resources included here and take a minute to answer our online survey. The Office of Cancer Clinical Proteomics Research is part of the U.S. Department of Health and Human Services at the National Institutes of Health, National Cancer Institute. For more information about OCCPR, please visit www.proteomics.cancer.gov. For general information about cancer or clinical trials, please call 1-800-4